The next topic is about gene mutations. So what are gene mutation? Mutation refers to a change in the molecular structure of a gene or the physical structure of a chromosome. So where do mutations occur? So we have two destinations or two places where the mutations occur. First is somatic mutation. When we say somatic, again, it occurs in body cells. So somatic mutations takes place in non-reproductive cells. Many kinds of somatic mutations have no obvious effect on an organism because genetically normal body cells are able to compensate for the mutated cells because our somatic cells have the regeneration capability. So, nakukuha niyang mag-heal itself. But then, during defects in the chromosome pairs, doon mas nakikita or na-physical express yung mga mutation. Some somatic mutations affect or has an effect during cell division. Because during cell division, the cells or the somatic cells are dividing uncontrollably. Mabilis yung pag-divide ng somatic cells. That's why it is the cause of many forms of cancer. Okay, the next one is about germline mutations. So, it occurs in gametes. So, kung yung isa sa body cells, germline mutation occurs in gametes or the sex cells. It is passed on to an organism's progeny sa mga susunod na henerasyon. So, future generations will carry the mutation. So, that's why it's called as germline mutations. So, what kind of mutations exist? We have mutations that are categorized by the length of the nucleotide sequences they affect. So, we have two kinds. First is the gene-level mutation, and the second one is chromosomal mutations. So, dun muna tayo sa gene-level mutation. So, in gene-level mutation, the changes are in short stretches of nucleotides. So, maliliit lang or maiiksing nucleotides, maiiksing codons lang, ilang codons lang yung naaapektuhan dito sa gene-level mutation. So, this affect the specific genes that provide instructions for various functional molecules, including proteins, in which it changes these molecules and has an impact on any number of organisms' physical characteristics. Okay, so hindi ganun kalala yung gene level mutation kapag konti lang yung codon na naapektuhan niya. We have here first the different categories or under ng gene level mutation. We have insertion, deletion, frame shift mutation, base substitution, and then base substitution has 3 subcategories. So, yun muna yung unahin natin, yung base substitution. So, the first one under gene level mutation is base substitution. Base substitution is the simplest type of gene level mutation. It involves swapping of one nucleotide for another during DNA replication. So, in base substitution, only one codon is affected. Remember that in one codon, we have 3 nucleotides. Even though there's only one codon that is affected, it can have an impact in protein production. Because, diba, kapag yung codon na translate natin siya, or nagkaroon siya ng anticodon in transcription, after transcription, it will be translated into amino acid. So, kapag nagbago yung transcription, mababago din yung amino acids. And then, it has 3 subcategories. The first one is missense mutation. Second is nonsense mutation. And the third one is silent mutation. Okay, so what is missense mutation? Missense mutation, the altered codon leads to the insertion of an incorrect amino acid during transcription. So, we have here a DNA strand which is adenine. So, lahat yon adenine. And this is a codon because it has 3 nucleotides. The anticodon, DNA into RNA, will become adenine pairs with uracil. So, uracil silang tatlo. What is the amino acid produced? So, the amino acid produced here is phenylalanine. So, we have PHE. So, meron tayo dyang phenylalanine. Pero ano nangyayari sa missense mutation? Yung isa daw nating nucleotide, napapalitan siya, nasaswap sa isang to another nucleotide. Example, napalitan siya ng cytosine. So, mababago din kung ano yung transcribed. Okay? So, yung transcribed na natin will be guanine. And then, ano pang mababago sa kanya? Mababago din yung amino acid. It will become UUG or uracil, and guanine will become the amino acid leucine. Okay, leucine. So, anong nangyari? Incorrect yung ating codon, incorrect yung anticodon, and incorrect din yung amino acid. So, from kanina, phenylalanine, naging leucine. So, that is missense mutation. Next is nonsense mutation. Okay, so the next one is nonsense mutation. Ano naman yung nonsense mutation? Altered codon prematurely terminates synthesis of a protein molecule. So, in order to create a protein, we need to have 20 amino acids. Meaning, ilang set ng codon yon. Ibig sabihin, kailangan din natin ng 20 set ng codons in order to produce a protein molecule. Okay, so dito sa nonsense mutation, I have here an example. 3 sets, the first 3 set of amino acid, 3 sets of codon. So I have here for the DNA strand, thymine, adenine, cytosine, adenine, cytosine, guanine, guanine, cytosine, adenine. When we transcribe it to an RNA strand, so it will be adenine, uracil, guanine, cytosine, cytosine, guanine, uracil. And when we translate it into an amino acid, so for the first one, we always start with AUG. So this is a correct amino acid, which is met or methionine. Next one, we have here another amino acid, UGC. UGC is for the amino acid cysteine, and then we have CGU for the amino acid arginine, ARG. So what happens in nonsense mutation? One nucleotide, if one nucleotide is changed, for example, dito nangyari yung mutation. Naging thymine instead of guanine. Okay, so ano mangyayari? Mapapalitan din ito, yung kanyang RNA, mapapalitan din siya ng transcription, magiging UGA. Okay, UGA. And then, mapapalitan na rin siya ng amino acid. It will be translated into what amino acid? Nakalagay dun sa codon chart natin, UGA stands for stop. Okay, so what will happen to the protein synthesis? Kauumpisa pa lang niya dito sa methionine, nag-start pa lang dyan sa methionine. Pagdating dito sa second nating set, nagkaroon na tayo agad ng stop. Meaning, ma-stop na yung synthesis natin ng amino acids. Ma-stop na yung paggawa natin ng protein molecule. So, baliwala na yung mga sumunod na set ng codon or set of amino acids. Okay, so yun yung nonsense mutation. Kapag prematurely terminated yung ating set of amino acid. Nagkaroon agad siya ng stop. Okay, so that is nonsense mutation. Okay, so the next subcategory of base substitution is silent mutation. Silent mutation is when an altered codon codes for the same amino acid as the unaltered codon. So, ano namang ibig sabihin nun? We have here, for example, again, TAC, CCA, AAA to be transcribed into RNA, AUG, GGU, and UUU, and translated to amino acid. Again, we have the starting codon, which is methionine. Next, this one, GGU is for glycine, and UUU for phenylalanine. Okay, so what happens in silent mutation? Example, here meron na naman tayong isang nucleotide na nag-mutate. CCA becomes CCG. Adenine is replaced by guanine. That is the mutation. Okay, anong mangyayari? Mapapalitan ulit yung ating RNA. So, guanine will become cytosine, GGC. Okay, so in our codon chart, kanina ang ating amino acid is glycine. Ngayon, pag tinignan natin sa codon chart, yung meaning na ng codon na GGC is also glycine. Okay, glycine pa rin siya, hindi nabago. So, yun yung silent mutation. Nabago yung composition natin sa codon, but then the amino acid does not change. That is what we call as silent mutation. Okay, next, after base substitution, also under the gene level mutation, we have insertion, deletion, and frame shift mutation. So, during insertion mutation, it occurs when an extra nucleotide is added to the DNA strand during replication. This can happen when the replicating strand has slips or wrinkles. So, makikita natin dito yung dalawang strand ng DNA. So, this happens during unwinding. Ang nangyayari kasi dun sa unwinding, di ba, ina-unwind siya ng helicase, hindi siya na-unwind ng mabuti. Okay, so ang nangyayari, nagkakaroon tayo ng tinatawag na slippage or wrinkles. So, ito, wrinkled yung part dito sa replicating strand. Ang nangyayari, nagkakaroon tayo ng insertion ng isang nucleotide. So, nakikita nyo dito, ito yung original sequence, and then, dun sa may wrinkle or dun sa may slippage, nag-i-insert ng isang panibagong nucleotide. Okay, so mamaya explain ko tong frame shift. Kasi yung insertion and deletion, nakaka-apekto siya dun sa tinatawag na frame shift mutation. The next one is deletion. So, in deletion, syempre, nadi-delete naman. Kaya lang dito, doon naman nadi-delete dun sa ating template strand, imbes na dun sa replicating strand. So, in deletion mutation, it occurs when a wrinkle forms on the DNA base strand and subsequently causes a nucleotide to be omitted. Okay, so doon naman sa wrinkle niya, natatanggalan naman tayo ng isang nucleotide, kaya siya tinawag na deletion. Okay, so anong kinalaman niya doon sa frame shift mutation? So, doon sa frame shift mutation, it is the complete alteration of the amino acid sequence of a protein occurs during translation because ribosomes read the mRNA strand in codons. So, di ba dito sa insertion natin, nagkaroon tayo ng may na-insert na isang nucleotide. So, anong mangyayari dun sa codons? So, dito, imbes na ang basahin niya is TAA as a codon, CTG na set of 3. Ayan. So, anong mangyayari dun sa insertion? TAA, yun pa rin siya. Pero dahil nagkaroon tayo dito ng may na-insert, iba na yung mababasa sa kanya. CCT. So, may iba yung reading frame na tinatawag. Okay, yung framing, yung set natin ng codon, naiiba siya. Yun yung tinatawag na frame shift mutation. Nagbabago yung translation ng ating amino acid because there is an insertion of a nucleotide or there is a deletion of a nucleotide. So, nababago yung set. Imbes na CTG siya noong una, naging CCT. Hanggang sa umipod na yung ating reading frame na by 3's. Ganun din sa deletion. So, kung ang na-read niya dati ay CTG, tas na-translate niya sa amino acid, no nagkaroon tayo ng deletion na wala yung thymine. So, ang basa na ngayon sa reading frame niya ng codon ay CGC. Okay, so nababago din yung translation doon ng amino acid. So, if the number of bases was removed from or inserted into the segment of the DNA, it is not in the multiples of 3. The reading frame will change. So, yun yung sa frame shift mutation. Umiipod siya, so naiiba yung nasa set of 3. What causes these mutations? Ayan. So, meron pa pala tayong isang kind of mutation. We have chromosomal mutations. Ayan. So, what are chromosomal mutations? If gene-level mutation happens in short stretches of nucleotides, dito naman sa chromosomal mutation, mutations that alter longer stretches of nucleotides. Sometimes multiple genes up to the entire chromosome. So, dito, mahaba yung naapektuhang nucleotides and ayun, kaya tayo nagkakaroon ng chromosomal defects kasi yung buong chromosome na yun ay nagkaroon ng mutation. Okay, so what causes these mutations? So, number 1 is it is caused by, it may be a spontaneous mutation. What is spontaneous mutation? It happens during the replication. Yung tinatawag natin kanina na slippage of the DNA strand, hindi maayos yung pagkaka-unwind ng enzyme na helicase. So, nagkakaroon tayo ng slippage or wrinkles doon sa ating DNA strand, causing for the nucleotides to have an extra nucleotide that is insertion and to omit a nucleotide that is deletion. So, that is spontaneous mutation. So, pagkakamali siya doon mismo sa gene. So, the excess flexibility of the DNA strand and subsequent mispairing of bases. Ayun yung spontaneous mutation. While the second one is environmental exposure. It is because of certain chemicals. It may be because of UV radiation or the ultraviolet radiation. And those are called as external agents of genetic change, also known as mutagens. So, yung mutagens natin are certain chemicals and yun, radiation. Mostly because of radioactive elements. Yung nagkakos naman doon ng mutation in terms of environmental exposure. So, what are the consequences of mutations? First, we have a source of genetic diversity. So, pag nag-mutate ang isang species, ibang species na siya. So, there are differences, may variations. Minsan kagandahan siya, and minsan naman, it brings harm. So, ano yung it brings harm? So, it causes illnesses and diseases, especially in chromosomal defects that later on I will discuss. And then, the next one, it is somehow beneficial because some of the animals, they mutate in order for them to adapt to their environment. So, those are the consequences of mutation. It became a source of genetic diversity, it caused illnesses and diseases, and somehow beneficial for some species due to adaptation.